Hello and welcome everyone in another episode of Researchers Celebrity. And today we have Victor with us as our researcher celebrity. Victor has recently joined University in Houston as an assistant professor. And welcome, Victor, for sharing your thing. And now I would like to welcome you also with the thing that if you can start how you started or when you thought that you want to be a researcher. Up over to you, Victor. Welcome.
Hi, Raja. Thank you so much for the invitation. So that's a very good question. I would start from the bachelor. So I first started doing biology. So I studied biology in Spain, where I'm originally from, and specifically in Valencia, which is the Mediterranean coast south of Barcelona. So I did biology there, and I just started biology thinking that I wanting to teach. So my first idea was to be a teacher, right? But then, I started first year, second year, and then we started doing some research in the lab. And I thought, wow, I can do this. And no one told me that, I can do research and I can contribute, right, with new knowledge. And there was when the spark came, like about third, fourth year of my biology degree, and I said, "Hey, I wanna do this. I wanna explore more this pathway."
Mm-hmm. So from there, how it ended up being in Houston?
Yeah, it's a long way, but I'll try to be brief. So I did my biology degree, as I said, in Spain. I also did my master in Spain. And after the master, I got into a PhD program in the University of Valencia, also in Spain. So I spent almost, at least 12 years in Valencia since I was 18 to 30, more or less, where I did my bachelor, master, and PhD. And then when I finished the PhD, I just had a talk from from Emmanuel Charpentier in Valencia. So she came and she explained us, around maybe 2015 or something like this. She came and explained about the CRISPR technology, which is, for people who didn't hear about that, is a tool that allows you to do genome editing in almost any organism. So I found that tool was very interesting and could be, really useful for the future as it is being now. And I decided that I wanted to learn that technique. And then I started looking for groups that were using this technique, and I found a group or two groups in University of California, San Diego, Ethan Beer and Valentino Gens, who are using CRISPR applied for mosquito population control.
Mm-hmm.
Also, San Diego is very sunny, as Valencia is. So I thought, okay, this is the perfect place to me to go. So I went to San Diego, did my postdoc from 2017 to 2022, so almost 5 years. And then I started looking for faculty positions while I was a postdoc, and had this opportunity coming here to Houston to UTHealth Science Center and start my independent research program, where I am now, and I started 2 months ago.
Okay, nice. That's good to hear your brief journey. So now, for our audience, Victor just said a name, Emmanuel Carpenter. She is the one who recently got Nobel Prize shared with Jennifer Doudna for her work with CRISPR. So when you said that you heard that talk from the Nobel laureate firsthand, what was the thing that you wanted to, because CRISPR can be used in so many ways, and you chose the group with who are working with mosquitoes. So it was a decision which you wanted to work with mosquitoes, or it was a thing that because it is translational, or it was a personal preference?
Honestly, I was more interested about the tool and the technique per se. So I was pretty open to go into any lab using CRISPR. So I wouldn't mind, going to a lab, for example, doing human diseases, working in cell culture, or other kind of model or yeast. So I wouldn't mind like any model. Was more the technique per se. But these groups are working with mosquitoes, but also with flies, right? So they use flies and then they translate the technologies that they develop in flies, they translate it to mosquitoes, Ethan Beer and Valentino. So I did my PhD using flies to model human diseases. So I already had experience with flies, and I thought could be kind of a sweet transition where I could learn CRISPR while working with the same model that I already worked in Spain. So that could make me, feel, more comfortable and having that transition, less, less, I would say, aggressive, right? So because moving from Spain to the US, also you have the culture shock, right? So there's a lot of things going on there. So I just wanted to minimize the change, right?
Very good. That was very intelligent, actually. So, would you like to share some important incidences what were the experiences when you were doing research in Spain and when you moved to California, San Diego University, University of California, San Diego? What was the basic differences in science which you felt as a researcher?
Well, the main biggest difference is the amount of resources. That's the first one, right? So in Spain, there's not a lot of funding opportunities for research. So people struggles a lot, with their fellowships, their contracts. The salaries are not, super high. You go from, for example, like during my PhD, I had 1 year contract, so every year needs to renew, right? And when I came to the US, they were already offering me 3 to 5 years contract. So I wouldn't need to be worried at least during 3 or 5 years, which also gives you, calm to work and just focus on research. So that was the main difference scientific wise, the resources. And also, which was pretty, surprising, is also the society. So when you go and people ask you, "Oh, what do you do?" So people also, knows more in general about science. It seems they appreciate it more. So I would say also the education here is that in general, the population also, they are super convinced that science is important, even if they don't understand everything, right? But they know that's important. So they really appreciate your work, which I think, at least in our country, in Spain, it doesn't. The message doesn't, arrive to the general population. So they don't really know how science could be important. I mean, they can imagine that it's good for health and the stuff, but they don't really know what's going on.
Okay. So this brings us to a very important question. When you communicate your research to a layman, like layman is a term which people just normally use, but for me, it's like the one who's not from your field. Like if you are explaining your biology to a physicist or, some chemist or some others, because they are still allied. But if you are explaining it to a mathematician or an economic expert,
Yeah.
how easy or how difficult you feel to convey?
I think I had some good training with my parents, right? So when my parents ask me, "What are you doing?" Right? So I already have the general audience, someone who is not from the field. So, when people ask me, "What are you doing?" Since it is related with mosquitoes, people know
Yes.
mosquito, right? Because, mosquitoes bite everyone. So everyone got some bites of mosquitoes at some point, and they know how annoying they are, right? So they know that they transmit diseases. So people in that specific field, they know a bit, but usually what I say is that I use, tools to engineer mosquito population so we can make mosquitoes resistant to some kind of parasites. So in that way, when they bite an infected person, they cannot transmit it because we introduced a new trait into that mosquito, so they are protected. It's like they are vaccinated, right, against the parasite, and we can do that in the lab, or we can also eliminate mosquito population. So if you eliminate a mosquito population, then they don't have the chance of biting and transmitting the disease. So that's kind of the explanation that I do. I don't go in detail about CRISPR or any terminology, right? So it's basically using genetics to control mosquito populations and help reduce the impact of vector-borne diseases.
Nice. This was like, I think this is something which I will suggest to all the viewers and audience. As Victor said, talking to your parents, probably they are your first listeners who can train you well, because nowadays communicating science is becoming so important that communication in science itself is a branch where people learn how to talk, how to express their science, like complicated science, because you cannot use a lot of jargons to a common person because they don't understand. But if you go with an attitude, as Victor said, like mosquitoes, everyone knows it, flies, or something which people can correlate with, it makes your work easy to tell them what you are doing and what your bigger aim is. Coming back to aim and objectives, Victor, when you see research allowed in lab and when it goes to translational, like when you are taking it to the field, what is the biggest burden you think, or biggest gap, I will say, you think that from lab to field, where it is lacking?
What it is, sorry?
Because people do, like for transgenic in mosquitoes, so many people are doing in the lab. There are so many groups around the world. But when it comes to go to the field, what is the reason why there is a distant gap?
Well, I think first of all is all the regulatory policies that we need to go through that we don't really know, right? So because we are trying to do science, right? So I think that's a huge step that we need to go through. I mean, I'm not saying that we are really, every person working in the lab has something ready to apply in the field. I think there is still work to do to have something ready. But I would say the regulatory policies are a huge step that we need to do through. And I think when you say genetically modified, is something that also is kind of scares people a bit. I think also part of that is that I was, thinking about this weekend, talking to my wife about that. Like, if you think about all the series in Netflix about scientists and the stuff, most of them are about scientists doing something bad, like doing transgenics that can, kill the population or like human population, right? So scientists doing something that is bad for the population. So that's also part of the message that you mentioned that is important to communicate, that we scientists wants to do something for the for the humanity, right? Not doing anything. But I mean, in the world you find all kinds of persons, but in general, we are doing something for good, right? And I think there is also a part of communication, as you said, where people need to understand that, because when you say genetically modified, then people get scared. But when you explain them, hey, that, for example, that the insulin, how the insulin is obtained, that we use bacteria that are modified, and because they are helpful, because logistically is easier. If you put some examples about how genetics is being used already, even people don't know it, right? So they start also understanding that word is just a terminology that can be genetically modified and can be safe. So that will be something that people, when they listen genetically modified, they totally go to, oh, that's bad, that's dangerous, right? But it's not always the case. So I think that's the two things I see. First, all the steps that we need to go through, and second, also communication, as you said. I think it's important because when we are doing some kind of intervention, it's important the public, opinion, right?
Yes, absolutely.
And they need to understand in order to to have better, well thought, decisions, right?
Yes, absolutely. So now, so this is about the public engagement in your research. If we take a step back and we think about ourselves, because you pointed out very well that all these Netflix series, all these movies, this says that, okay, scientists are doing something bad. Most of them are. But recently, what the entire world has experienced during the COVID time, all scientists, all the researchers broke all the history of a vaccine development. They just changed it to the level that everyone started appreciating research. And one of the reason of having researcher celebrity is that it's not just one person who does it. It's the entire community who's
Mhm.
there working 24/7 for public good. There might be some experiments which don't go the way they want, but that is less than 0.001%, which
Mhm.
means the motive here is to work towards better human health. And I think now everyone can appreciate that, okay, the COVID vaccine came so fast, so quick, and saved so many lives.
Yeah, but then here I think that's a very good example. Now I think people started appreciating, right? But
Yes.
if you take this step back, you also know that at the beginning that we developed the vaccine like so fast, as you said. Then
Mm-hmm.
a lot of people say, "Oh, how can you develop a vaccine in 1 year if
Yes.
usually it takes 10 or 15 or
Yes.
20 years, right?" So
Yes.
there is always that skeptic uh,
Absolutely. Yeah.
part. Then you need to explain, hey, this vaccine, there's people that was already working with RNA vaccines for other
Yes.
kinds of diseases. So there's a
Yes.
lot of knowledge
Yes.
there. So they applied all that knowledge to this specific case, right?
Mm-hmm.
You can
Yes.
sequence genomes of the virus. This
Mm-hmm.
you couldn't do it 20, 30 years ago, right? As
Mm-hmm.
fast as you can, you couldn't. Now
Yeah.
you can get the virus one day, next day you have the genome, right? So there's a lot of things that we learned, we improved, and this because now we can do some stuff fast. In this case, we were lucky, and people, being able to travel and to go out of home, people realize how important that was, because it was very tough. Stay at home, you know, eliminate all your social life. We are social animals, right?
Mm-hmm.
And that was very tough for all of us.
Yes. So here I would like to make a note to all our audience that research is something which is not an overnight job. As Victor said, there were scientists doing their research for ages, for decades. Be it technology development, like sequencing a virus now is like the entire genome, you can do it in a day and you can do all of that. But it was not true 1 decade before. All these developments come together. That's when all the researchers think and work towards better human health. This is one thing. Second thing is when now something is available. So as Victor, you mentioned about just sequencing the genomes and all, but there are now during the COVID time, people even started doing the repurposing of drug. So that drug was there, but they thought it can be used for this or not.
Mhm.
So there was a certain amount of research already done, which was there, and now it is about utilization it differently. So, Victor, when you see doing research, spending almost 2 decades in research now, including your bachelor's, how do you feel that the fundamental research and the translational research, how does they go and how much you can connect with them when you feel that one of the things which you are doing is basically only in the lab, which has not either direct or immediate implementation for human health, whereas when you are doing one.
So how you convince people about that, about the two
How
different?
do you feel about it? Like when you are
I
doing the
feel like
basic?
just my opinion.
Yes.
Yeah, I think it's a very good question, but also a tough one because, uh, we are humans, so we want solutions and we want results, right?
Mm-hmm.
So, being patient is difficult, even for us that are scientists, right? You
Mm-hmm.
want something now.
Mm-hmm.
So if you develop a vaccine, right, people is happy.
Mm-hmm.
But if you're studying, the biology of the RNA to
Mm-hmm.
understand how you can use that information to then, produce a efficient vaccine,
Mm-hmm.
that, is not as, impressive, right?
Mm-hmm.
So when you explain to someone, they are not going to be interested about
Mm-hmm.
that. And I think that again is with related with the public communication.
Mm-hmm.
We and governments and all the funds go towards something that is important for society, right? So
Yes.
sometimes we don't realize that we need that basic research. But for example, CRISPR
Mm-hmm.
is a perfect example, about basic stuff, right? So I will use now this time to explain a bit about that because so I also will use that because one Spanish scientist was involved in all this
Mm-hmm.
CRISPR history, and not many people don't know about
Know
him.
that. Yes.
So that is a very good example, right?
Mm-hmm.
So CRISPR, right, is the immune system of bacteria, right? Same
Yes.
as we have an immune system, bacteria they also have the immune system against virus.
Mm-hmm.
So this came when a Spanish scientist he was going to some different beach in Spain, right, in Alicante, so that's the Mediterranean coast,
Mm-hmm.
and he was studying the different microorganisms
Mm-hmm.
in high saline, spots. So
Mm-hmm.
places with high salinity,
Mm-hmm.
he was going there, taking samples, and then sequence the genomes, right?
Mm-hmm.
So he realized that they have a specific pattern in the sequence of different microorganisms,
Mm-hmm.
what was weird and interesting at the same time, because that
Mm-hmm.
pattern is why all these microorganisms have the same specific pattern in their
Yes.
genomes.
Mm-hmm.
Why? Right? So he realized that that was important for the immune system of the bacteria to protect their cells against the virus. He
Mm-hmm.
was not the one, you know, having that amazing, ability of saying, "Oh, I could use this for genome editing,"
Yes.
which is the reason why Jennifer Doudna and Emmanuel Charpentier got the Nobel. They got the Nobel for genome editing, right?
Yes.
But that basic research, which was just going, take some water samples,
Mm-hmm.
sequence the bacteria, just curiosity, how the genomes of this bacteria look
Mm-hmm.
like, brought us to this revolutionary tool about CRISPR. So this scientist from Spain saw this, and then the other people, for example, Dona and Charpentier, say, hey, if this system, can attack virus genomes,
Mm-hmm.
right? When bacteria gets infected, why we cannot, move this system and use it for a global genome editing tool? And that's what they did. So you have that example, but
Mm-hmm.
I'm pretty sure if you ask people,
Mm-hmm.
I mean, I would say at least more than 50% for sure don't know about the existence of these previous studies. I'm
Absolutely.
I'm pretty sure. So
Yeah.
that would be one example just to reinforce the fact that we need basic research. The problem is that that's not as popular or
Mm-hmm.
famous, so people maybe don't appreciate it as much.
So here we come to 2 points. The first point is, as you said, it is about glamour. The fundamental research is not that glamorous, not fancy. People are not known for their contributions. But this is even true for the scientist, as you mentioned about the great contribution of the French scientists in there, that not many of them know. But what is more important is that somebody has done that work. They have invested their time. They have done all the research, what was required, and how I see research as it's a puzzle, not a single puzzle. It's like there are so many pieces of this puzzle. You collect them, you make one picture, and this one picture is a part of another bigger picture. And it goes on and on and on. And that's why there is no end to research. You do something today, someone will use your research for betterment, new transformation.
Yeah.
And then who knows that when, as you said, when doctor was like the scientist was doing all those analysis, he might have not thought that this will be used for genome editing. The information will be the base for translational science, and then somebody will get Nobel on that.
Yeah. Now, sorry, I will say his name in case some people want to look it up. So he's called Mojica. Mojica, and he's in Alicante. He's a professor there still, and he still does science. But people don't know about him. I mean, he got recognized in Europe with a lot of prizes and the stuff. But I mean, I'm not saying that he deserves like the Nobel. Okay, I think it's fair what happened.
So here, the beauty of this researcher celebrity conversation is all your thoughts are your thought. Neither we are making claims nor we are denying any. So
but yeah, I like to mention that because that's also important. That I think he did a very huge job and amazing discoveries. But the Nobel was a different part. Was for genome editing because the application is what made the revolution. You can apply genome editing for human diseases, for agriculture, for pest control. The broad application of the genome editing discovery that these ladies did. I mean, that's amazing.
So this brings us to another point of collaborations in research. How do you feel the collaboration increases or reduces the pace of research?
The pace. Oh, I think, of course, collaborations, are usually for good because when you get different expertise together, you always can think bigger, right? And you always can have thoughts together that maybe you cannot have alone, right? Because if you explain your science to another scientist from another field, they will see gaps in the science that you can fill with your expertise. And you couldn't see that gaps because your mind is focused in another part, right? So I think I always say that if you take two scientists with different backgrounds and you put them in a room for 2 hours, I'm sure something good comes out from there. So I think that's always good. Barrier that I see there is that also is even within the scientist community, we have different vocabularies, right? And we have different expertise. So sometimes it's also difficult to communicate and to make scientists understand that we can get something good from someone who maybe we think, oh no, that's so far from my field. But then you realize that it's not as far when you really think deeply about it. So yeah, I mean, I saw it during my scientific careers. Almost all my publications are collaboration and involves at least 2 groups. So I would say, I think except one, all of them, involve different groups and made that research, being rich in ideas and new concepts and, and new applications.
Nice. So here, the bullet point is collaboration is the key. You should be always open for collaboration. And as Victor said, because you are so much involved in your field that you sometimes don't see the gap which a person coming from another field brings in. And those questions actually improves your research, and the collaboration makes the impact even bigger.
Again, another good example, sorry, that we keep talking about CRISPR, but Jennifer Doudna and Emmanuel Charpentier is also a good example. Jennifer Doudna
Yes, absolutely.
is in UC Berkeley in California, and
Mm-hmm.
Emmanuel Charpentier was in France. She was in Europe. So they met each other in a conference. They started working together, and then is when they, described the CRISPR as genome editing tool in 2012.
Yes.
10 years now.
Yes. So, talking of on the decades of CRISPR collaboration, as you said, from 2012 to 2022, you pointed out about the collaboration happening at conferences. How important a researcher should see, because when I have seen it, and I was probably one of them at some point of time, that when the new PhD students or graduate students or it be master students, when they go for conferences, they see that as a fun trip. Most of them. So
they see it as us, sorry.
So they see it as a fun trip. They go there and have fun.
Country. Yeah.
But what here is a thing which everyone should consider, that when you are going for a conference, always make note there are people who you are interested to talk to. They can guide you, they can help you, and they are there, which can be the milestones in your journey also. What are your thoughts, Victor, about conferences, meetings, how a researcher should approach that?
Yeah, I think can be both. Can be fun and can be useful as well, right? So yeah, I think conferences are always good because it brings inspiration for your research, right? Because I think you sitting in your office, writing your grants, and living in your bubble, it's tough to get new ideas. So it's I would say that the conference is a good spot for inspiration, where you see people presenting different technologies. Then you can think, oh, I could use the technology, as we were saying, to fill a gap in my field. What is something that we couldn't do, but now with this technology that this person is presenting here, I could talk to him and say, hey, do you think we could apply this? And then you can know about the details of that technology. And it's always good, the conference, because also something that COVID showed us is that, at least in my opinion, the best thing is to have the meetings in person, right? Because you can approach, you can talk, you can go for a coffee. These kind of recordings are very good when you're at home, for kind of as educational tool. But if you want to increase your networking, you want to improve your research, I think like conferences in person, are key to bring inspiration and improve your research.
Nice. So we talked about research, we talked about conferences. I believe that everyone can see the passion and the dedication you brings towards your research. What is the one thing, if you think that because of researcher, you have to compromise in life? What is that one thing?
What I am compromising life? I don't think I'm compromising anything. Why?
Okay. So because I will tell you the reason why people say this. Yeah.
Okay.
People say the work-life balance in researchers.
Okay.
Life. Be it family time, be it with friends.
Yeah. So I think I did pretty well in that kind of work-life balance, at least in Spain and Europe in general. I think there is a good work-life balance. It is true that here maybe, that is not, as good. I think that's also kind of cultural, right? Here people, is super hardworking. I'm not saying that in other countries are not, but I would say that here, if they need to put more hours into their work, they are okay. Instead, in Spain, if you tell people to do a lot of more hours every day, they can do it, but they will also be thinking about the family part,
Mm-hmm.
thinking about going with friends, right? But I think that could be something cultural. So coming here, I'm trying to, to keep that work-life balance, which I will say that my postdoc was very good.
Mm-hmm.
Also, that's a good question, because I always like to talk about this. People think that the work-life balance, it's also important the people that you have around. Okay.
Yes. Yes.
So, my postdoc advisor, he was the first one, kind of encouraging to get a coffee, right?
Mm-hmm.
And to build team and
Mm-hmm.
to build transparency. So if we need to go for a coffee, it's fine. Then we come, we work. So, finding this flexibility, having a mentor that trusts you, one day you can go home soon if you don't have anything to do.
Mm-hmm.
Another day, if you need to stay more, you stay more, right?
Mm-hmm.
So I have that kind of mentality, which I also want to implement in my team.
Yeah.
I trust people, and they can manage their time.
Mm-hmm.
So I think that's also part of everyone being able to make their own decision, right?
Yes,
So
absolutely.
and also being surrounded by good people. So for me, I would say the toughest part, and I want to be honest, the work-life balance was worst during the transition from postdoc to faculty.
Mm-hmm.
That transition was tough, and and I spent a lot of time, doing applications, interview, thinking about how to do strong applications. So that I would say was worst in terms of time involved in that
Mm-hmm.
process compared
Mm-hmm.
to my PhD or my postdoc.
Okay. So here I would like to tell all of our audience that Victor is a genuine person you can contact anytime for the help, and the hurdles he has moved and excelled. Probably you can get some help from him. So we are at the edge, on the edge of our conversation, Victor. So with
Okay.
we would like to thank you on behalf of Empowering Science Foundation, and we wish you very good luck. And now what we want is few key points you want to suggest for the budding researcher, the researcher who are doing their postdoc and looking for faculty.
Okay. So I will start a bit, I will take a step back. So when you start your postdoc, my suggestions would be, first, choose a good mentor or a person who trusts you and who gives you flexibility also within your research. So start thinking about your projects soon. And while you are doing projects from your lab, also being able to have some time, 10%, 20% of your time, thinking about how you can distinguish from your mentor. I think that's very important from the beginning. Second, I would say start writing soon. So, convey to your mentor, hey, I want to improve my writing skills. Because when you start applications and writing grants, so it's something that you need to train, to get trained. So I think that's the most important thing during the postdoc.
Mm-hmm.
And the third one is be kind with everyone, because you don't know where your collaborator, where your letter of support can be, right? So be kind in general, be nice, and be open. And with that three things, I think you have a good chance
Mm-hmm.
of finding a position. And of course, when you start looking, then
Mm-hmm.
I would say be very calm and patient,
Mm-hmm.
because it takes time. So take your time to prepare the applications
Mm-hmm.
and contact people who is in the committee for
Mm-hmm.
the faculty positions before even submitting, just
Mm-hmm.
to give an overview of your work. And
Mm-hmm.
then from the emails, you can see the excitement, right? If they
Mm-hmm.
say, oh yes, apply, maybe they are busy, doesn't mean that they don't want your research. But
Mm-hmm.
sometimes you can see the opposite. If they think that your research is very interesting, you are going to see that, right?
Yeah.
So they would say, oh yeah, that's very important, it's very relevant for our search, so please apply, right? So that I would say is important. Focus and apply. Not, I mean, at least that's my opinion. And don't apply everywhere. So apply to places where your research makes sense.
Nice. Thank you very, very much, Victor. This was enlightening, and I believe all of our viewers will be benefited with that. With this, we will take leave for now, and we will see you all in another episode of Researchers Celebrities.